Developmental Biology, Unit 5 of CSIR NET Life Science syllabus. Many a times students will ask, what are the important topics of this particular unit that we need to prepare for our exam, and from where should we prepare it? Hello biotechnicans, today I will give you a brief concept about what all you should study from this unit that will help you to answer at least 80 to 90% of the questions that come in CSIR exam. If you talk about the syllabus for this unit, these are the 5 subunits that you will get, and here they are marked according to the importance, the weightage of the questions that will come. The first subunit being Basics of Development, it is all basic and foundation concepts that you will need it in the later parts of the syllabus. Some might think that these concepts are needed only to understand the later topics, but no, this is a very major subunit from this unit from which questions will come directly. Terms and terminologies like potency, commitment, specification, induction, competence, determination, differentiation, morphogenetic gradients, fate map, cell fate, cell lineage. There are direct questions from these terms and terminologies many a times which has come repeatedly in CSIR syllabus exam. Stem cells and genomic equivalence, there are not much questions regarding it, and from unit 5 there are no questions from imprinting. However, you can get a related question in genetics, but till now in unit 5 there has been no questions from this area. Cytoplasmic determinants, another important factor in the different type of specifications that we have. Autonomous specification, what is the use of cytoplasmic, what is the significance of cytoplasmic determinants in autonomous specification. A little bit of history also, like how we use the mutants and transgenics in analysis of development, who were the pioneer scientists whose experiments have paved the milestones of understanding the subject. So a little bit throwback also comes in exam. Okay. Then we have this subunit B, which is Gametogenesis, fertilization and early development. Here the most important parts are the cleavage divisions. First of all, the zygote formation. In that also fertilization is important, where we have medium important, where the sperm egg recognition in mammals are asked, and different concepts based on that I will come into detail. Then the cleavage divisions, which are the cell divisions in early embryo. Blastula formation, after cleavage divisions, the ball of cells, solid ball of cells which you get. How it is formed in different organisms. The gastrulation phase that leads to the formation of germ layers in triploblastic animals. These all also come in exam. Okay. If you talk about in plants, establishment of symmetry in plants is not that important. However, embryogenesis, seed formation, germination, sometimes question is there. But embryogenesis in plant and fertilization in plants is also important, where the double fertilization questions are also asked, and embryo structure is also asked. What are the layers that you get, right? Then we have unit C, subunit C, where the Drosophila axis and pattern formation is extremely important, followed by even amphibia and chick. Sometimes comparison questions are also asked. Development in amphibia, development in chick, or development in chick and development in mammals. Vulva development in Caenorhabditis elegans, which is the nematode. This is also an important topic. Limb development and regeneration. Of late, they have started to give a lot of questions from this area. That's why recently it has become a very important topic. Differentiation of neurons, it is another topic. And neurulation, which you can say, which is also kind of important. Then we have this post-embryonic development, which is larval formation, metamorphosis, environmental regulation of normal development. Till now, questions have not yet come from this area, so you can easily skip this part. Okay. And cell aggregation and differentiation in dictyostelium, it is a semi-important topic. maybe 2-3 exams once there will be a question on this topic. Okay. Sex determination also follows the same pattern. From plant development, there is organization of shoot and apical meristem, leaf development and phyllotaxy, transition to flowering and floral meristem. These are semi-important, but then again I should tell you that in development biology, animal development questions are much more. Obviously, the syllabus is also covering majority of the portions. And even in the questions also, you will get 80% to 90% from animal development and only maybe 10%, maybe one part B question and one part C question from plant development. That too, it is not fixed always. Okay. And what is important in plant, most of the times question come from this only, that is floral development in Arabidopsis. Subunit E is actually a part of unit 4, which is programmed cell death. However, you can study a little bit on aging and senescence as well, because sometimes they might give a question maybe in 2 to 3 years there may be one question. So again I'll tell it as a medium important topic. Let's dive into the must topics covered from these individual subunits. As I said, from the first subunit you will have the important terms and terminologies like potency, commitment, induction, competence, cytoplasmic determinant, morphogenetic gradient. You have to understand the in-depth concepts. For example, if a cell has to be induced, then what are the ways it can be induced? So here one important unit also comes into picture, which is cell signaling. You have to know cell signaling and also cell communication, how the cells will adhere with each other, the different adherence junctions and the signaling pathways. This go hand in hand with unit 5 developmental biology. Okay. And if a cell has to receive the signal, then the cell must have the competence. Now, what are the types of induction that you have? Regional specificity, genetic specificity, or you can say conditional induction, instructional induction. So these are the different in-depth concepts you have to understand with reference to the different developmental processes. And you should be able to differentiate. Differentiating questions come here. Mainly you will be getting true or false statements, most of which are opposite and contrasting statements. You have to find out what is the difference, such as specification, differentiation and determination. Types of specification like autonomous, conditional, regulative, with examples. For example, in tunicates you will get autonomous, in sea urchin you will get conditional development. At which stage will you get? Every detail, attention to detail is something which you have to pay attention in this unit. Okay. The basic terms and terminologies of development: potency, commitment, and as I just said, induction, competence, cytoplasmic determinants and morphogenetic gradients. Practice as many questions as you can from this particular area. Okay. What are the questions asked from here? As I said, you will get definition and concept based, most of which are true or false statements, right? And differences between the types of specifications. In autonomous, how the cells get their cell fate. In conditional, how they get their cell fate. If you remove a small part of the embryo from autonomous, then what will be the result? If you remove a small part in conditional development organism, what will be the result? So these all combinations methods you should study, and especially experimental result based questions. See here, unit 5 is a very theoretical type of subject in which you have to memorize a lot, right? But yes, if you have a certain understanding of the pattern of development, then it will not be so difficult for you. And mostly here the questions will be experimental also. If you transplant this, or if you mutate this gene, then what will be the effect? So whenever you are preparing for this subject, you should study from these lines beforehand, so that you don't get any unexpected surprise questions. And as I mentioned, from this particular subunit, historical perspectives also you should keep in mind. For example, Rauch's experiment, Dresch's experiment, there has been direct questions which have come based on this experiment, like what will be the conclusion. Okay. While we are on this particular topic, experimental based questions, historical based questions, whenever you are getting any question, please do not have any preconception in your mind regarding what result the scientists have got or what the standard experiment results are. Do not assume that and try to answer this question. Maybe they will give you a different scenario, the experiment will have a little different kind of setting, and that's why you have to look at every experiment based question or historical based question from a fresh mind, keeping your concepts in the background. Okay, so probably they are asking, this is your experiment, what will be the result? Now if you are taking some standard experiment and pasting the results over there, then you're not going to get the right answer. This is where students will go wrong, and you have to practice it like that. Coming to the second subunit, which is gametogenesis, questions are not that common from this topic. The stages and chromosome number, you don't have to study that much, you can ignore, but to understand fertilization it is better, and also from unit 8 or unit 2 cell cycle you might require this to study this. Fertilization is important, yes, especially the blocks to polyspermy, slow block and fast block to polyspermy. Again the details like terms like cortical reaction, acrosomal reaction, and even the fast block, which is opening of sodium channels, graph based questions, concept behind this, everything should be crystal clear. The fusion of egg and sperm in model organisms like sea urchin, amphibian and mammals are asked mainly. Okay, so you will not get the fertilization part from any other model organism, so you can study only from these three. Capacitation is another concept from mammalian fertilization. Questions have come from this area as well. Then when the zygote is formed, the different types of eggs and what different cleavage patterns these eggs will undergo. Like we have telolecithal, mesolecithal, alecithal, depending on the amount of yolk and also on the distribution of yolk, there is different classification, and specifically the example of organisms like telolecithal egg is present in birds, then in fishes and in reptiles. Okay, then what kind of fishes? In bony fishes it is present. So like that you have to be very clear about the different type of cleavage pattern and also in which it takes place along with the fate map. So this type of egg, if you have, then after fertilization what type of embryo it will form, and in that embryo which part of the embryo is supposed to give what type of structure. So fate maps also you must know. Then the gastrulation part, which is the main chunk of portion of developmental biology. It might look a simple subtopic, but it covers many organisms. So you have to study the gastrulation process, the cell movements and axis formation, how the dorsal ventral, that is back side, front side of the body is determined, how the anterior posterior axis is determined in the body, like how it is decided which side is head, which side is tail. So there are some signaling processes involved in here, you have to know about all of them. And also the proximal distal, if your limb outgrows from the body, then what decides which is going to be the part which is closer to your body and which is going away out of the body. So everything in model organisms of sea urchin, C. elegans, amphibians, zebrafish, chick and mammals. You have to study all of this. In amphibians, it is very important. Topics like spemen organizer, role of beta-catenin, BMP, nodal, and in chick you have primitive streak, Henson's node. These are very interesting and very important topics. In sea urchin you have to know the role of micromeres. So what are the questions asked from here? Different graph based questions on slow block, fast block, like what composition, what happens to the concentration of sodium ion or different proteins, receptor proteins, like that. Arranging the events of fertilization, where the chemoattraction happens first or acrosomal reaction happens first. Attention to detail is important. Direct questions about molecules involved. As I said, BMP, nodal, beta-catenin. Even the pathway based questions are very important. Okay, types of eggs and various cleavage patterns. You must know the classification of eggs based on amount of yolk and distribution of yolk. And what are the types and subtypes of cleavage? For example, if you have alecithal, that is without yolk, then it will undergo holoblastic cleavage, means it will divide completely. The egg will divide completely. If you have telolecithal or centrolecithal, which have some sufficient amount of yolk, then they will divide incomplete. That is called as meroblastic. So these subtypes are important. For example, you have this cleavage division, one example from Gilbert book. Under isolecithal also you have different types of cleavage, and as you can see here some representative organisms, these are what are very important. That's why you must memorize this properly. You can get direct questions. Match the following questions are very common from this area. Okay, so diagram based questions, identification of types of egg or the cleavage pattern. They will give you either diagram, identify what type of egg or what type of cleavage pattern. They can also give you fate map, and as I said, match the following questions, which type of cleavage and in what type of representative organism. Coming to early development in model organism, you have to study the cleavage, division, pattern and gastrulation in all of this representative organism. Zebrafish is not mentioned in your syllabus and question is not that common. Maybe in 2-3 years once they will give a question. However, sea urchin is also not mentioned in your syllabus, but sea urchin comes a lot. Okay, at least in 1-2 years it will definitely come question, sea urchin. Most important is amphibians, chicks and Drosophila. So these three are the major organisms in which development you should study properly. Focus on the important cells, genes, molecules involved in the process and also the signaling pathway that are involved. Axis formation, dorsal, ventral, anterior, posterior, organizer regions, what type of molecules they will secrete, then mechanisms which are involved in that, pattern formation, whether it will give directly pattern, there will be the sequential arrangement or morphogen based pattern formation. So these are the question asked, most important sections of the syllabus. All types of questions can come like transplantation, experiment based, mutation and analysis, all the genes which are involved, if you mutate any of them, then what will be the result, concept and process based and also diagram based, where you have to identify different molecules of the signal pathway. Okay, signaling pathways, the important ones in development perspective is sonic hedgehog, then delta notch pathway, beta catenin WNT pathway, and in that you should focus on the ligand, receptors, secondary messengers, transcription factors, sequence of events, and what will be the activators and inhibitors. So how can they be activated or inhibited? These all focus should be there. And obviously you must also study the other signaling processes like receptor tyrosine kinase, GPCR. Anyways, those are part of the unit 4. So these are some hedgehog pathways, as you can see. In absence of ligand, in presence of ligand, what are the different activations which are happening? These all concepts are very important. Okay. Then must topics to be covered from unit 5C. They are developmental, Drosophila developmental stages, the different types of maternal and zygotic genes which are involved with the detailed process of function. And this is a very scoring topic because definitely questions will be there from some part of it, either the genes or the organogenesis part, organogenesis, limb formation, role of Hox genes, then in limb formation, AER, apical ectodermal ridge, fibroblast growth factor, then regeneration in amphibians, neurulation, neural development, sex determination, vulva development. Sometimes eye-lens induction questions also come, and as I say, sometimes CAM signaling also will come. Apart from this, all signaling pathways have to be prepared very well, right? So in limb formation and regeneration, you have to study the tetrapod limb development in vertebrates, the roles of different Hox genes, and experimental data-based questions, limb regeneration. In neurulation, you can study the types, primary, secondary neurulation, the different stages of that, how the BMP signaling is done, and how the folding is done, what is notochord, the notochord axis formation, what will give the ventral signal, what will be the dorsal signal, the molecule regions and the genes involved. And neurulation questions have come for comparison in chicks and mammals, like neurulation in chicks and neurulation in mammals. They can ask comparison questions. Okay, vulva development, every time question will be there, so you can take it as a sure shot topic. What are the cells involved? Anchor cell is there which gives the signal to vulval precursor cells. So you should know the cell names, the anchor cell, what type of signaling events will be there. Mutational analysis, if you mutate, overexpress, or if you do a negative mutation, then what will happen? Sex determination, basis of male and female determination, how it happens in mammals, comparison to that how it happens in Drosophila. Dosage compensation and role of molecules such as sex lethal, transformation and double sex. All these molecules roles you must be aware of. Also alternate splicing, because that is what affects the male and female sex determination in Drosophila. From plant, must topic covered. From plant development, you should focus on double fertilization, leaf development, meristems and their transition to flowering, and also the complete floral development along with ABC model, with emphasis on the genes which are involved in Arabidopsis and Antirrhinum. So you can study the genes which are involved in embryogenesis in Arabidopsis, formation of ram, sam, root apical meristem, shoot apical meristem. Then this floral organ identity genes, every time question will be there, that is flowering in Arabidopsis. And sometimes there are questions on vernalization in flowering. Okay. So double fertilization, leaf development, meristems and the transition to flowering, ABC model, you can focus on all this from plant development. Okay. What are the standard reference books from animal development? You have the Bible of Development, which is Gilbert book. Everything which you need to study are very clearly and with proper diagrams, it is already mentioned in Gilbert. Then you can also refer Wolpert. This is another book, Louis Wolpert. And one more is there, J.M.W. Slack. So better you can refer Gilbert only. But along with that, if you want, you can also see these concepts also. In plants, you can study this Linden book for plant development, cellular basis. You will not need all the concepts, only study the important ones. Then Hopkins, plant development of Hopkins. And even this Charles Beck, which is plant structure and development. Okay, so here the focus will be in anatomy part. What are the topics to be studied from online resources? So you have this vulva development, which you can study from something called as Worm book, because you will not get so much detail in books as such. Okay, cell aggregation in Dictyostelium discoideum, you will have to see some review articles for that, as you will not find much proper information in books. And some plant developmental topics like shoot and root development, leaf development, phyllotaxy, if you don't find the books, because it might be hard, so then you can refer from online resources, but go for only standard resources, that is NCBI or Science Direct review articles. Okay. You can also revise from Concept Wheel book of Biotechnica, which is any particular topic and what are the different points under that topic. So every point of the topic is color coded, which is very helpful in recollecting during exam. Like BMP, it is a type BMP inhibitor, extremely important in amphibians. Questions have come. So certain parts of the embryo induce the ectoderm to become epidermal tissue. So what are the functions and what do this organizer tissues act by secreting molecules that blocks this induction? So what will be inhibitor, what will be activator, what are their function, what is the significance, all these points are covered. Okay. We have the concept table books, as I said, from unit 5A, the differences between the different type of specification, autonomous, conditional, syncytial, all these differences, as you can see, it is in one sheet. Okay, so each of this can be one sentence in a true or false question. So studying from them and revising from them mainly, it will be very beneficial. Conceptica Light, it is about the diagrams and the flowcharts. You can refer from this also. That was all from my end. Thank you everyone.